All right, thanks. Thank you everyone for joining. So today's seminar speaker is Dr. Emily Puckett from not too far down the road, the University of Memphis. Dr. Puckett earned a bachelor's degree in botany from North Carolina State University and a master's in plant physiology from State University of New York College of Environmental Science and Forestry, and a PhD in biology from the University of Missouri. She also completed postdoctoral work at Fordham University and Michigan State University before joining the Department of Biological Sciences as an assistant professor at the University of Memphis in 2018. Dr. Puckett's research incorporates phylogeography, conservation genetics, evolution, and comparative genomics and population genetics. She is interested in the evolutionary history of mammals, in particular, the brown rat and the black bear. I don't want to get that confused. She has over 20 publications and book chapters on these topics and has made significant contributions to the understanding of the origin of invasive species, phylogeographic patterns in mammals, and on using phylogeography, I might be pronouncing that wrong, to aid in species conservation. So Dr. Puckett, thank you for joining us today and welcome.
Thanks everyone for attending my talk today. And I just want to tell you about how rats conquered the globe and the phylogeographic history of Rattus Norvegicus. So the first part of my talk asks, how did brown rats go from here, the Mongolian steppe, to here, almost every city in the world? This specifically is New York City, and I'll mainly be talking about work I did as a postdoc with Jason Munchie South at Fordham University in the Bronx. So what is phylogeography? It's the field of study that elucidates the processes governing the geographic distribution of genealogical lineages within or among species. So my main study species is the American black bear. And so usually when I give this talk and tell people what phylogeography is, I tell them, well, I never get to talk about the brown bear phylogeography. So here's just a little taste of that species. So in this example, we see in the colored polygons on the map, the range distribution of the contemporary range distribution of the brown bear or Ursus arctos. And then we can link that to, this is a mitogenome tree, and we see the different clades represented in that species. And here I'm going to highlight 3A in blue, 1A in purple, and 1B in orange. So there are multiple lines of evidence that have identified where the glacial refugia were in Europe during the last glacial maximum, 18 to 22,000 years ago. And so when we put these pieces of information together, we know where those refugial habitats were. We understand which genetic lineages are found in the contemporary space, as well as where those genetic lineages are not only just in the refugial areas, but also across that geography. We can then infer those routes of range expansion out of the glacial refuges that then expanded across the contemporary landscape. And so in this example, we see that 1A has spread from the Iberian Peninsula across Europe. 1B was much more isolated. Of course, there are big mountains in this region. And then 3A expanded up into Fennoscandia. So we can put all of this information together, and that's really what phylogeography is doing. So phylogeography can answer a number of questions, including what are the barriers to dispersal for a species? What's the divergence times for a species, and how does that relate to those glacial refugia? Where are the contact zones? And whether this is hybridization between species or admixture within species, we can find those areas of secondary contact. The location of glacial refugia, right? The phylogeography would be another line of evidence that we can use, although geologic evidence is the strongest for finding those areas of glacial refuges. What are the routes of range expansion? So that's kind of the example I just did. And then very importantly, what's the spread of infectious disease? So a quick outline of my talk, I just want to start with Rattus and Rattus norvegicus, and really remembering that they are a species and they have a natural history, as well as an evolutionary history. Talk about specific work on the range expansion around the globe of Rattus norvegicus, and then end with thinking about brown rats as a medical model and how much of that natural diversity is in the sequence lines. So before I dive into Rattus norvegicus specifically, it's worth stepping back to consider the genus Rattus. So there are 64 species within the genus, and they are native to Eastern Asia and the Australopalpine biogeographic region. So I'm showing in this figure the most complete Rattus phylogeny from Rowe and colleagues, and it only has 27 taxa represented, or about 40% of the named species. So morphological data has been used to place those other species within clades within the phylogeny, but full and even partial mitochondrial sequences are not available for most Rattus taxa, right? It's just like I think we think it's an incredibly well-studied group, and it's not. Within this phylogeny, I've used the purple colors on the tips to highlight taxa that can live on artificial habitats. In total, 27 of the 64 species are thought to be able to use artificial habitats. And the International Union for the Conservation of Nature, or IUCN, defines artificial substrates as croplands, rural gardens, and urban areas. The ability to use artificial habitats has likely evolved multiple times, or it's been lost multiple times. So I didn't want to color these internal nodes in this tree to suggest where these traits may have evolved, given the incomplete nature of this tree. I have a sense just from the topology and how many of these can exist on artificial habitats, that for this Asian clade, it's been overrepresented by species that are found in artificial habitats or urban environments, and that the ones that can't use those have not been sampled and therefore are unable to be included in the tree. So most research on the phylogeography and evolutionary history of rats has focused on the 4 commensal species. Rattus excellens, or the Pacific rat, is the first one, the first big commensal. And it is most famous for being introduced across the Polynesian islands with the spread of people. So these animals were likely a food source that was easily transported by boat, and therefore from this putative natal range on this island is spread in multiple directions across the Pacific and then into those Polynesian islands by the Polynesian people. Rattus tanazumi, the Asian house rat, and Rattus rattus, the black rat, are 2 additional commensal species. I'm showing them together because they're part of a cluster called the Rattus rattus species complex, where are they species, are they not, has not fully been delimited at this point, but they don't tend to have overlapping ranges, with tanazumi having dominance in Eastern Asia. And then Rattus rattus is native to India and spread via human trade towards Europe, Africa, and then from Europe over to North and South America. But the star of today's show is Rattus norvegicus and the brown rat, and it's one of the most successful human commensals. So why study the phylogeography of brown rats? So brown rats are one of the most successful human commensals, house mouse potentially rivaling it as the most successful. These species use our food resources, access to water, and possibly use our built environments for shelter, and potentially the human control of their natural predators. So humans are giving these species a lot and very often not having any harm in return, although that's not always true. If rats need these resources for humans, we may understand their presence as an indicator of city size or human economies. And archaeologists are interested to link the presence of rats to what that may say about settlements that are only found as archaeological digs now. So the phylogeography can inform understanding those routes of dispersal. Rats are also a globally invasive species, and this is of conservation concern, particularly for threatened and endangered bird populations. So rats will eat the eggs, and given both the high growth potential of rats and birds' slower population growth, this can have strong conservation effects for the persistence of those populations. So how does phylogeography help here? It's that when we know the genetic signature of rats in an area where an eradication program is being planned, we can use that for future monitoring to understand, was the eradication program potentially less effective and that rat population size has been grown after the eradication effort, or have new migrants settled and colonized this area that is meant to be protected. So rats do carry and transmit diseases to humans, so monitoring them as a vector occurs globally. And it was this research community that we tapped into for our global sampling efforts. And finally, they are the second most used medical model. I'm sure this audience knows that very well behind house mouse. And so another reason to study their natural diversity. Again, they're a species, right? They have a natural history. And so rats prefer to live in earthen burrows. They live in colonies with this really interesting hierarchical social structure. They have very high natural mortality. About 90% of the population turns over annually, but they also have very high reproduction. So females can have 6 to 11 pups per litter, and they can breed 2 to 4 times annually. We don't have a great sense of how latitudinal variation changes the number of times that they can breed annually, although we do think that it might have some variability across the range. So I just wanted to mention that there's kind of a limited knowledge of both the ecology and evolution of this genus, but where we do have a good understanding are really with these 4 commensal species. So now I want to move on to the part of my talk about Rattus norvegicus range expansion. So the questions that we asked in this project are, what routes did rats take? And when I say rats, I mean brown rat, to spread around the globe. And when did this range expansion occur? So my postdoc mentor was able to tap into this global network of biologists, many of them studying rats for zoonotic disease, monitoring, to build a global collection of brown rat samples. So we looked at 76 cities or farms and initially obtained 314 samples. We genotyped these samples using double digest restriction-associated DNA sequencing, or dd-RADseq. And this is a reduced representation genotyping approach, which resulted in 32,000 genome-wide SNPs for us. We further go on to, after we looked at that diversity, we selected specific samples for whole genome sequencing, and we got the whole genome sequencing and for 12 samples, and then used some analysis techniques, MSMC, as well as FastSimCol to look at that data.
Emily,
so I'm going to show.
Emily, what was your N per city?
It ranged. It could be as low as 1 or as high as about 15.
Okay, thanks.
Yeah. So we're going to show some evolutionary clustering results. And if you're not used to this data, I just want to walk you through like what you're going to see. So you're really just looking at stack bar graphs where every column is an individual sample from a single animal. And the color is representative of the clustering level, or the cluster that proportion of genetic ancestry is being assigned to. So in this toy example, sample 1 is 100% from the pink group, and sample 2 is 100% from the purple group, but sample 3 is looking like an F1 with 50% pink and 50% purple. So the other way we could think of this is instead of at the per sample level, at the population level. And so here, if I average the amount of pink and purple genetic diversity, I've got 50% pink and 50% purple in this example. And so here's our data. We start by splitting the data into 2 clusters. And what became very clear very fast was that there was this Asian associated cluster. And again, we think that they're from endemic to Mongolia and northern China, as well as a non-Asian cluster that we see around most of the rest of the world, including in Europe, North America, South America, Africa, and New Zealand. We do see bits of blue ancestry in predominantly brown samples, as well as brown ancestry in predominantly blue samples. And this could either represent unsorted ancestry or admixture, where diverged populations are coming back into secondary contact. Because individuals may, like, depending on place, I kind of think of some of these differently, right? So New Zealand, I think this is admixture. Some of this, I think this is just unsorted genetic variation. So as we add levels of clustering, going from, and K equals clustering, so adding 6 levels of clustering instead of 2, we start to see kind of a population tree form. So here we see this breakout of this unique Aleutian archipelago cluster, as well as these gold samples are from San Diego, but they have that background from being predominantly of Asian ancestry. And this, we can see similar things in the non-Asian ancestry, where some North American and Northern Europe samples are separating, as well as this Haida Gwaii population breaks out very early. So the best supported number of clusters was 26, which is on this bottom row. But when you dive into that pattern, I would argue that the high number of clusters was driven by the software being able to separate individual cities and assign them as unique evolutionary clusters. But we weren't interested in that specific question. We wanted to think about rats at a higher hierarchical level of organization. So not the individual population or city level, but really the lineage. We still think that the cities are interesting, and I think it shows a lot about how little movement and also the bottlenecking that occurs as a population gets to a new locale. But ultimately, the rest of my talk will really be either at this K equals 13 level or the K equals 6 level. So how does that clustering data plot at each city location? And so this is what the data on a map looks like. And I am showing it at the K equals 13 level. So there are 2 primary patterns I'd like to draw your attention to. First is that the Asian cluster is mostly found in Asia, right? So right here. However, the second place that it is found is Western North America. And so this was actually a surprising result for us, because ultimately we show that North America has been colonized both from the East and the West at different times. Secondly, the light blue from Western Europe is found throughout North and South America, as well as Africa. And this is due to the introduction of rats all over the world by the European colonizers that use ships to move people and goods all over the world. So I want to pause our global expansion journey for just a few minutes to talk about what we did learn by focusing on rats within New York City. So here is that New York City data, and here I've just blown it up for you. So we were surprised at how homogeneous the ancestry components were across samples from the city. We expected a pattern more like on the right, and this is just totally made up data. But we thought we would see this because New York is this huge global city. It's got 3 airports, container ships going up the Hudson, container ships going up the East River, and how many cars and 18-wheelers passing over those bridges and those tunnels every single day. So we have 2 ideas behind why we didn't see what we expected. First, we didn't sample rats in New York City, we sampled rats in Manhattan. Thus, the airports are actually in Queens and Brooklyn and New Jersey. And so, the shipping ports are in Brooklyn and New Jersey as well. So it's not necessarily that those ports are in Manhattan per se, and that's where we did our sampling. The ports are actually further away geographically. Our second idea is that of priority effects. So rats are both territorial and have a well-organized social structure with both low and high caste individuals. Thus, an alternative hypothesis is that migrants may have been coming into New York and over time filling up this niche space. But that gene flow does not occur because at some point the urban niche has already been filled. In this model, even if there was admixture, so we do feel like we see admixture in our data, among the lineages that we found, that those could have occurred early during colonization, such that that new rat with a unique genotype comes in at the port, the niche is already filled, and there's just no space for it anymore, right? So all of the early genotypes had priority in establishing across the range. So we haven't tested this. To test it, we would really need to start monitoring from those ports in a transect-like path radiating out from the ports to see if migrants may be coming even if gene flow is not occurring. Our first analysis of rats in New York City had just those 10 samples that I just showed you. But we expanded this to 284 samples across the island. And those are being shown in the red points on this map. And again, we use dd-rad-seq SNP genotypes. Our first analysis confirmed that this is a single population. However, with this much data and this dense sampling, we were able to pick up what are called genetic neighborhoods, which are a hierarchical level of organization that you can also think of as an extended family group. You can see these neighborhoods in these black and white points in these maps, which we refer to as the downtown and uptown rats. And as we zoom in on any group, it will continue to break down, right? So genetic diversity is hierarchically structured. So if we look at these uptown rats, we can see that it breaks down into kind of a Harlem group, as well as a Midtown and upper Manhattan group. We thought that Midtown was a particularly interesting region in that we identified lower gene flow than expected in this area. So Midtown is between 59th and 14th Streets, and it's a very high affluence area with many of the businesses and tourist attractions that you have likely enjoyed when you've been to New York City. So this is where MoMA is, and Times Square and Broadway, right? So all of that is in Midtown. And so the field teams actually had a really difficult time catching rats here. And that's why we have such low density of sampling from this region. Thus, we think it's a barrier to gene flow because effective eradication efforts are going on in this high affluence region of Manhattan. But the big point I want to make about this, thinking about within city spatial structure, is relative to the extent of spatial autocorrelation in genetic relatedness. So we found that rats were related, like relatives, right, these genetic neighborhoods, up to about 1,400 meters. And that's about 0.87 miles, if that's easier for you to think about. Yet we saw the strongest relatedness was observed within 200 meters, or about 3 city blocks. Without putting GPS trackers on juveniles to directly estimate natal dispersal, the spatial autocorrelation analysis gives us an idea of how far dispersal events are within a dense urban environment. There's limited work in this area, but dispersal is, the work that has been done shows that dispersal distance is further in both suburban as well as rural habitats. We believe this is related to the high resource availability within the urban environment. And our results have implications for the extent that eradication programs would need to occur at. So if you're interested in eradicating rats on your block, you're really going to need to expand 2 blocks in every direction to manage that gene flow that is going to occur based on just natal dispersal. I see a chat, but I'm not reading it. Sorry.
Yeah, well, we can come back to the chat questions later.
Cool. All right. So switching back towards the global story, one big question is, what paths did rats take to go around the world? So to explore that, to explore the routes that rats took from their natal range around the world, we used a method called range expansion. And this compares the site frequency spectrum between 2 geographic locations in a pairwise manner and looks for the loss of alleles that we would expect to occur during a founder event. In this way, it can identify both source and sink populations. And I show directionality of that source using magenta and the sink in this gold. So things in this figure are moving from pink to gold. And the thickness of the line is proportional to the z-score only for the significant relationships, and non-significant relationships are not being shown in these figures. On the left panel, there is a broad trend where source populations appear in the north and sink populations tend to appear in the south, although we do see this sink population in the eastern region as well. And my interpretation of this was supportive of that hypothesis that Mongolia and northern China were the natal range for the species. There's still an open debate here as fossil evidence for the species is very scant, but we do see the range expansion genomic signatures as supportive of a northern to southern hypothesis. We next compared Southeast Asia with the modern day Middle East, and we did not see the expected pattern. So we thought that we would see source populations in Southeast Asia and sink populations in the Middle East. That's not quite what we saw. And I just want to highlight and not hide that that data is what it looks like. When we compare the modern day Middle East to Europe, we observed that the Middle East has a signature of being the source into places throughout Europe. From here, into radiating all over that continent. I'm not showing it here, but that pattern is also found between Europe and Southeast Asia, so that Southeast Asia does look like the source for the populations in Europe. We also see that there's this, right, so within Central Europe, we again see some radiation of potential Central Europe being a source for more distant populations out, including in Finno-Scandia, Great Britain, and the Iberian Peninsula. It's important not to overinterpret this data and only think that an overland expansion could have occurred, but instead understanding that there could have been ships from either of those places that are moving rats either to into ports on the Atlantic seaboard or on the Mediterranean, and that we just don't have those Greek, Italian, and a number of the Atlantic seaboard cities sampled.
So Emily, wouldn't this have happened during the pre-Yamnaya Celtic era? I mean, we're talking about just barely any shipping, Phoenician, pre-Phoenician. We're talking about people with carts and wagons dragging themselves, up to glaciers, I would think.
We think it's 1100s, 1200s is what our demographic models had. So, right, so not necessarily as late, but some of the archaeological evidence, so I have a colleague that argues that there's some weak archaeological evidence for rats in Italy in the mid 1400s, but that the stronger archaeological evidence for brown rats into Europe, right? So we know that the black rats had 2 waves into Europe that occurred before the brown rat got there.
Okay. So that's the distinction I'm missing, the brown rat versus black rat. So everything yes. Bjorn Curtain did, that was all black rat?
Can you, one more time?
Bjorn Curtain, the famous paleontologist who studied, he's studied basically everything in Palestine. Mm-hmm. That was all black rat, I assume then?
I think so, yeah.
Okay.
Yeah, and I'm sorry I didn't make that distinction. I'm only talking about the brown rat, and that the black rat was already in Europe when brown rats were moved in.
Cool.
Yeah. So when we look at North America, we observe those 2 independent colonizations that I mentioned earlier. So one colonization from Europe to the Atlantic seaboard. We think it was, right, so there's some more direct evidence that it was to the Atlantic seaboard, and that some of these significant relationships are really overland migration from the East Coast to the West Coast after they were introduced into North America. But then, let's see, a colonization we expected this given European colonization in the 1600s, but a second colonization did occur from Asia towards the Pacific seaboard, including into the Aleutian archipelago, which is down here. The oral history of rats onto the Aleutians says that this did not occur until the 1780s, and that it was Russian fur traders from Kamchatka that introduced rats to the Aleutians in the late 1700s while they were hunting seals for pelts that were sold across Russia and China. And our data has some light support that even these Aleutian rats do move towards, again, the more mainland sampling areas that we have. To further understand the demographic history of brown rats, we estimated their population size through time. So if you're not used to these plots, the x-axis is time since the present. So we're over here at the origin, and then years ago we're going backwards in time as we move from left to right on this figure. The y-axis is effective population size. And the input data is whole genome sequencing, where each different colored line that I'm showing represents either 1 or 2 animals, and that's going to be either 2 to 4 haplotypes. So we see 2 distinct patterns. The first is an older signature observed in China, Southeast Asia, and European samples. So effective population size begins to decline around 100,000 years ago, and then stops around 1,000 years ago and begins increasing in size again. So with these plots, you kind of always want to discount the last 2 to 3 time lags. And even if we like lop those off the graph, we do still see this uptick since about 1,000 years ago in population size, which kind of makes sense with what I'm saying, that as they're expanding their range, they've got more area to live in, and overall effective population size is going to increase. So thinking back to the natural history of rats, right, they're really similar to most mammals, right? So in a lot of these MSMC or PSMC plots, we do see that during the most recent glacial cycle, that population size has declined over time. The second signature we see is much different, and it's found in the Aleutian and Western North American samples. And this, right, so it doesn't even appear on the graph until around 50,000 years ago. And it does still have that steady decline through the LGM, but it just bottoms out, and we don't see that uptick in effective population size. So we think that this lineage is more limited, and that actually makes sense with the limited geographic areas in which we can find it. So if I take those clustering results and the range expansion results and the population size results, and I put them together, I ended up building a demographic model within FastSimCol. So the best fit model had a ghost population as the ancestral population, because we don't think that we really did sample the ancestral range. And then 3 branching points, one into Eastern China, one into Southeast Asia, and then the last towards, likely towards Eastern Russia. That ultimately is what we're finding on the Aleutians and Western North America. This analysis also got us towards our second main question about when these expansions occurred. And so we estimated that rats expanded into Southeast Asia.
Uh-oh. Uh-oh.
Let's hope Emily comes back.
Into Europe. I'm not super happy with the exact times that this model picked up, and I think that we would really need more whole genome sequences to get truly precise estimates on really any of these numbers. Although I do think that ballpark, we're probably in kind of a reasonable range. So I do just want to throw that out there as a caveat on this data. So to kind of sum up this section, we see that rats expanded southwards into Southeast Asia before making that big global dispersal. And we see a unique and deeply diverged lineage that colonized Western North America. And now I want to kind of shift gears a little bit and talk about how the natural diversity that we see in brown rats might relate to the medical models that many people here are using. So the current thinking is that brown rats have been domesticated 3 independent times, although the independence can also be questioned. So the earliest evidence for the commensal relationship between brown rats and humans occurs in central and northern China and has been dated to about 7,000 to 9,000 years ago. And so this evidence is, again, these archaeological settlements and the presence of, I believe it's rat mandibles within the profile of the settlement. So in terms of the domestication events, in Japan, they were domesticated for both pets as well as fancy rats, in which they were bred for these fancy coat color patterns. And we still see some of these patterns today. So here's the fully coated one, and here's the hooded, and a potentially albino, and then one with more spots. And so that domestication event is, depending on the records that are looked at, started sometime between the 1650s and the 1770s. And so it's thought to have been the earliest domestication event for rats, for brown rats. In Europe, the domestication started in the early 1800s, and rats were used for blood sports. So actually some of the samples from our global collection came from blood sports. So the dogs would go out to some area, and it's a rather violent death for the rat. So the dogs pick them up and they shake them until they die. And so that is still ongoing today. But initially, rats were domesticated to be used in this sporting event. In North America, we see the domestication for scientific studies. It's unclear if the colonies established for scientific studies came from already domesticated European stock. And the reason why it's unclear is that when we read the oral history, especially of the Wistar Institute, and Donaldson having these 4 pairs of albino rats, right? So the albino form is not common in nature, and there's at least some indication that when the European blood sporting events occurred, they would pull out the albinos and keep them separately and not let them go into the baiting pool. And so those may have been a source for the colonies established in the United States, although it's unclear. But it's also been suggested, especially with the Brown Norway line, that the members of the Wistar Institute went out into Philly, grabbed some rats, brought them into the lab, and domesticated some wild animals. So I want to focus the last part of my talk on what we've learned about the genomic diversity in a select number of rat strains and how that compares to natural diversity. So at the time that I worked on this project, Antonar and colleagues presented the most comprehensive genomic analysis of inbred rat strains. Since then, more rat strains have been sequenced, both as part of the hybrid rat diversity panel or even some of the work coming from your department. So several groups present numerous analyses of strain-specific genetic diversity, structural variants, and sweeps related to disease phenotypes. But I'm going to focus my work on Antar's analysis because I grabbed all his whole genome data a couple of years ago. So this group performed whole genome resequencing of 27 different inbred lines. And y'all would know better than I that I've seen reference to either 220 strains or even as many as 500 different strains of brown rats. But the point here is the same, that 27 lines might not be representative of all of the different strains that are available. So their phylogenetic tree does show some expected patterns of shared ancestry among the strains. So up here we see the Japanese Wistar-derived groups, and all the known Sprague-Dawley derived strains are clustering together and share a common ancestor. So the authors use the same data in a PCA and state that the first and second axes encompass 70% of the genomic variation within these 27 strains. And this analysis, it recapitulates parts of this phylogeny, pulling out Sprague-Dawley from some other strains and separating those Japanese Wistar-derived strains on the PC1 axis. It's worth noting here that both the Lewis and the WAG strains, let me see, here's Lewis and here's WAG, are known derivatives of Wistar stock. So even when it's saying, it kind of gets here, right? So Wistar Italy, Wistar United States, but that Wistar Institute really did have a huge effect on multiple of these clades and the genetic diversity within the strains. The point I'm trying to make is that the analysis, these types of analyses, both a perfectly bifurcating phylogenetic tree as well as the PCA, they're going to find differentiation in the input data, regardless of if the data is like has high differentiation or low differentiation. So what I did is I took the WGS data from the 27 inbred strains and pulled out those 32,000 dd-rad-seq loci to match our global diversity data set. And then I ran a PCA on that data. And I'm showing all of the inbred samples in black in this figure on the right. In this PCA, the Asian cluster is on the right side. So samples here and here. And the non-Asian cluster is predominantly on the left of PC1. PC3 separates out Western North America from the rest of the Asian clade. And also note that I projected the inbred strains into the PCA space of the global genetic diversity, and I didn't allow the inbred strains to influence the structure of the PCA space, because what the question I was asking is, where in the world are they from? So my hypothesis was that they would cluster with these light blue samples that are representative of Western Europe and North America, due to the oral history of domestication from those geographic regions. And that's not what we found. So we're seeing that the inbred samples are clustering, at least on PC1, closer to the Asian strains, and then even on PC3, not really pulling towards those light blue samples. I will draw attention to brown Norway, right? So this sample right here. And brown Norway is, of course, the strain used for the reference genome. And this was developed at the Wistar Institute in Philadelphia. And I think I said it a minute ago, the oral history is that they were grabbed outside in Philly, and that became its own independent colony. So I would argue that, yes, we see that way closer to those North American samples, but also still having a lot of shared ancestry with the other inbred strains. And so it's in a unique place in that PCA space.
Emily, this is Hao Chen. I have a question. What happens to PC2?
PC2, I just thought it was not informative for like really giving a lot of information. So if I show PC1 versus PC2, all of the black samples basically sit on top of the Asian samples, because PC2 is really pulling out these Haida-Gawai samples right here. They're so bottlenecked, they pull really hard on PC2. And so there was just like not a lot of information in this analysis.
I think Hal and I love this plot, which I've never seen before, because it puts BN in the center instead of at the periphery, which if you did it with the Antor data, BN would be off in the ozone somewhere about half a mile away.
It would.
BN is still half a mile away from the rest of the inbred rats.
Yeah, but who cares about those other inbred rats? Anyway?
Yeah, and BN has an interesting mitogenome too. So we'll see that in a sec. So what I would like to show you is how that nuclear genomic data is related. And we're going to do that instead of that clustering analysis, and we're going to use a co-ancestry heat map. And so what this is it's a sample by sample comparison of co-ancestry, or again, kind of an estimate of relatedness. So here we see that rat1 is being compared both to itself, to rat2, and to rat3, and that the color being shown on the heat map is related to that co-ancestry. So low co-ancestry is going to get these white or yellow colors, and high co-ancestry is going to get the brown colors. So here we see that rat1 has low relatedness to rats 2 and 3, but that rats 2 and 3 have high relatedness to each other. So we can look at this at an individual level, or we can look at it at a population level. And in this analysis, we see that rat 1 is the only individual from its population, where that rats 2 and 3 form a population together. And so here's my co-ancestry heat map for the global diversity plus the inbred rats. Again, white and yellow colors are going to mean low co-ancestry, where black and darker blue colors are going to mean high co-ancestry. We see the broad separation of the two main clusters that I talked about earlier. So here are the Asian samples, and here are the non-Asian samples. And in the co-ancestry heat map, we see that all 27 of the inbred rats cluster together right here, and that their closest geographic relationship is with that Western North American population represented by the samples we collected in San Diego County. They do show elevated co-ancestry with samples from Eastern China, but that's still, right, so right here, a little bit of elevation here, but still ultimately fairly low co-ancestry. So I have two big takeaways from this result.
Brown Norway be on there? It's not in the inbred cluster, is it?
It is.
Oh, it is? Okay.
Yeah, it's like right there.
Why isn't it closer? This is clustered in a sensible way. I'm just surprised we don't see it farther down on the diagonal. Since that's what the PCA would have suggested.
I agree with you. And I think it's that it's not as, I certainly can't say for certain, but I do suspect that there was some of that original Wistar ancestry, and it's not fully just Philly rats. It would be.
Could it be the genotyping platform is radically different? So you're using Antor for the 27 and you're using your own data for everybody else. So PC2 is probably technical, right?
Again, PC2 really pulls out Haida Gawai, which is a strongly bottlenecked island population. But yes, there could be some technical differences. I took all the data and mapped it to the BN6 reference genome, and then just did a pull down on the genotypes.
Okay.
So, so my two big takeaways are the currently sequenced inbred diversity is highly related to each other. And even given the oral history of these strains, specifically for lineages like Long Evans, which were not thought to share common ancestry with Wistar, we don't really see, or we see limited differentiation within the data. Secondly, I would like to argue that we need much better geographic sampling especially of Western North America, to understand how this lineage from San Diego might be represented. There is an alternative hypothesis that the rats that we sampled in San Diego share this high co-ancestry with the inbred strains, not because the rats in San Diego are somehow really unique, but maybe that they're former pet or lab rats that have escaped into the city. And so I do think that this needs much better geographic data to really hone in, although I would still argue that it has a stronger Asian signal than it does non-Asian signal. So all the work that I've shown thus far, both from Antonar and myself, focused on that nuclear genomic information. However, we could look at it from a mitogenome, maternally inherited perspective. And so this is a mitogenome network, and I've got those groupings from Antonar circled in my little key up here. So you see the blue Wistar Japan, here's the orange for brown Norway and Sprague-Dawley and the other strains. And when we look at the mitogenome diversity around the globe, we see that only 3 mitogenome clades, 14, 15, and 10, are associated with any inbred strains. The Wistar-derived lines are more closely associated with clade 15, and the Sprague-Dawley lines more associated with clade 14. Notably, both of these clades share ancestry patterns with samples from Asia, which does tend to match our nuclear data on that point. However, brown Norway has a clade 10 mitogenome. And I'm not showing it here, but in our Proc B paper, we show that clade 10 is really that European global expansion. So we see it in high frequency in Western Europe and almost exclusively in Eastern North America and South America and Africa. And so that is at least suggestive of that oral history, that at least some rats from one of those locales, like Philly, got into that brown Norway line. So these results again reiterate the limited mitogenome diversity encompassed within inbred strains, and that our geographic sampling is incomplete because we don't find clade 14 or clade 15, or we haven't found it thus far in nature. So to sum up this section, I would argue that geographic sampling is incomplete to fully understand strain origin, and that but also that there's limited natural diversity within those strains, right? I kind of expected a little more spotting all over that graph and to see how clustered it was. Again, maybe it's just because of what has been sequenced and was available on SRA, and maybe it's because of the true history of strain development. So I see several open questions. I also see I'm running out of time. Moving forwards, first, what adaptations facilitated the successful global invasion of the species? And we can come at this question from multiple different angles, including adaptations to the anthropogenic stressors or different climates, including their ability to live at high latitude. Second, what are the biotic and abiotic factors that limit their success into the tropics? Black rats outcompete brown rats at lower latitudes, whereas in temperate environments, brown rats outcompete black rats. We have a weak understanding of the fundamental niche for each species, as well as how that niche changes with latitude. And third, how much genetic diversity has been captured within inbred strains. I think the data support that a limited amount of genetic diversity has been captured, at least in the strains that have been sequenced, as I just mentioned. So with that, I would like to thank all of our sample collectors around the world. Special thanks to my postdoc advisor, Jason Munchie-South, and some key analysis contributors. And with that, I am happy to take your questions. Thank you so much.
Great, that was a very interesting talk. I see some questions in the chat, but I guess people can just go ahead and ask their questions in an orderly fashion.
Great talk, Emily. So is it possible to estimate for the inbred brown Norway from MCWI? I know there are a few MCWI people, RGD people on the call right now. Is it possible to estimate that particular line, how much of its genetic ancestry is coming from different populations?
It should be. Both the clustering analysis and the co-ancestry heat map, right? If we because it is an all by all comparison in those co-ancestry heat maps, we should be able to see some level of relatedness. I've never tried to do specifically what you're trying to do, but I'm pretty sure it's possible.
I see Anne is on one of the participants. Anne, any thoughts? You've been doing this forever.
So repeat what you were saying. I'm trying to understand if the MCWI is different from other.
No, I just want to know the history, the ancestry of MCWI strain in terms of other rats that are available.
Yeah, well, I mean, they're SS, they were N SSN, right? HSD. So they were at Harlan's. I mean, just by looking at the nomenclature, you can sort of get a history of where they came from, right?
Yeah, that's a history of when it started to be inbred. What I'm interested in, like,
yeah. Well, I've always said the brown Norway. Well, I'm glad to see what you're showing, because I always said the BN is more like a vole than a rat because it was so different from the other inbreds, but it's actually more representative of the rat than our inbreds. So, yeah, I guess you could probably get an idea just looking at the different BNs, like the what is it, the BNLX? How far away is that from the sequence BN?
Yeah, I just wanted to say I found this talk fascinating. I've just finished reading David Reich's book about human population. I'm looking for the book right now about human admixture, reticulated evolution, Neanderthals, little people, et cetera, et cetera. The inferences are very, very hard to make. The actual history goes back more like 200,000 years, and people don't have records of ships sailing into Manhattan and Montreal and Quebec. So I would encourage you strongly to do all 64 members of the genus. I mean, that would be fantastic, right? You said 64 members of the genus right now. So let's say if you could get us a spleen from each one of 64, or better yet, 4 spleens from each of 64. Oh my God, I would be like a rat in heaven, and Hal would be like a pig in heaven, and we get all telomere to telomere, and it would be a mind-boggling evolutionary biology experiment with real hardcore data, and we could see to what extent it's really just a directed acyclic graph, which it's not. It's going to be highly reticulated. And only in a species like rat, and maybe only in a species like the recent, brown rat demographics in history, could you really look at that admixture. And how gave us a genome biology paper to look at while you were talking. I kind of wonder, it's very tempting to talk about selection driving this, but for me, it's much more attractive just to think of it as drift and pure chance, because those NE numbers you showed, those are pathetic numbers. That makes Finland and Iceland look sad, right? We're talking, 500. So, you can imagine 2 really weary rats getting off a boat in Manhattan and saying, oh, do we have the energy to do it? And they're saying, no, we're too tired.
Those numbers do change. So, and again, I do think the data that went into the demographic model, of course, everyone could use more data, have more accuracy, more precision on their estimates. But one analysis that we did was Manhattan only. And I think the demographic model and the effective population sizes we estimated there, right? So what I showed you was kind of at the lineage level, but we could, of course, zoom into a very specific population. I think we hit, when we get more data, we hit a really strong estimate that when you think of effective population is so frequently about 5 to 10% of census size, and we think census size in Manhattan is 250,000, and we were hitting about 25,000 in the effective population size demographic estimates. We published that in an Ecology and Evolution article on cranial morphology shape change in New York City rats. And so yes, I agree that those numbers that I presented were weak, but they've got that weird scale dependency, right? So maybe the Aleutians are correct and China's not, or Western Europe is probably not. So lots of stuff going on with needing better or more samples.
So are you planning on collecting rats from other Western North American cities, LA, San Francisco?
Possibly. I really need, a lot of my focus has been on black bears right now in terms of starting up my lab, but I do think that there's a lot of interesting stuff to do here, and that a Ring of Fire project, really from Japan through Korea, Russia, Alaska, and then dropping down into the West Coast of North America would, I just think it's really biogeographically interesting. Because even those divergence times that I showed, we have to think of, they could have split in Russia and just been chilling in some glacial refugia and then got moved over in the late 1700s, right? They're not truly representative of when they got to North America. So I think that a much better sampling would be needed to use genetic only methods. But I think that archaeologists are going to get us much further into understanding if rats got here in anything earlier than just on ships because of trade.
That'd be interesting.
Yep.
Is there
And
any
so I am
efforts?
interested in talking to anyone who might have collaborators or might want to work on thinking about, expanding that global diversity data set and how it could possibly help other researchers who utilize rats in their research. I'm sorry, I interrupted you.
Yeah, the same pattern happened in mouse genetics about 40 years ago when Eva Eicher started, and other people started wild capturing mice in Southeast Asia everywhere, and then bringing them to the Jackson Laboratory and spending the 10 years it's required to turn them into an inbred strain. It increased the diversity in mousedom or inbred strain by probably about tenfold. I will say that so Howe and teams, the number of variants we have already is close to 10 million with relatively good maths of, 0.1 and above. So I'm not distressed. We're even the rats we have right now are more diverse than basically all human populations because we were so severely bottlenecked ourselves. So it's not a bad model, but it would be sensible just to generate 5 or 10 diverse inbred strains of rat. And you have the data now to pick those pretty much, right?
Probably, yeah.
It's probably about $25,000 to do a single rat, and then it needs to have a home. And unfortunately, rat doesn't have the same kind of home that we have for mouse, but MCW is probably it. And I think we have a lot of momentum right now in the rat community. We're trying to rescue rats for real genetics instead of just trying to replicate rat for CRISPR-Cas9, which I can see doing that, but that seems like a very limited opportunity. For me, the big benefit of these two other members of the supraprimate superorder is to use them for experimental precision medicine. And that means we have to deal with hundreds of inbred strains that encompass a lot of diversity. Then we have to be willing to make the F1 hybrids that are not inbred so that we really recover normal type genomes and see if we can predict outcomes to warfarin. Who's going to be resistant to warfarin? Who's going to be not resistant, et cetera.
Aren't rats already resistant to warfarin?
Yeah, it's a tricky drug for humans, that's for sure. And apparently a lethal drug for most rats.
So is there any efforts in Memphis to collect rats?
Not right now, no.
We can start one.
Okay.
Why start in New York City, right? We're here in Memphis.
We have them for sure.
Yeah, we need our ancestry diversity. We don't want to focus on those coastal types. We're much more interested in the Mid-South.
That's great.
I agree. I think having across the US would be really informative
Mhm.
instead of just the coasts.
Yeah,
Well,
I say that being in flyover land here in Milwaukee.
but I also think that we have some limited data that, again, we think that they went from the east westward, but also we have some rats in our sampling from Pigeon Forge, and they had higher proportions of that western signature. Again, maybe that's representative of pet rats or lab rats. But yeah, it seems like the western has also moved eastward. And so we might have some sort of gradients of admixture across the United States.
So Emily, just to point out the opportunity for you, I would say the group that's listening to you and probably twice as many people are really you're basically looking at the rat genome community right now. There's not much of it, but it's much bigger than it was 5 years ago, or even it's actually much bigger than it was in the heyday of the STAR Consortium, frankly. What we don't have is anybody that knows anything, to the best of my knowledge, about population genetics. So you could be in a driver's seat and guide us, because we're just a bunch of medical researchers, right? Poor medical researchers. And we could sure use some population geneticists to hold our hand occasionally and tell us what kind of NSF type applications would be practical. Because NSF is very interested in broader evolutionary population genetics issues. NIH will go to sleep on that kind of topic. But I think there's a great opportunity for you to take a leadership role with us. And we can certainly provide you lots and lots of sequence data. And I'm quite curious about if you were able to get 64 samples of spleen, we would find the wherewithal to get long read telomere to telomeres done. I'm pretty much committed to doing that.
Gotcha. I mean, yeah. Look forward to continuing to talk to y'all.
So along those lines, I had a question. So when you sample from a location, how do you know that you've sampled enough individuals to capture like the diversity in that population?
We really don't. So, right, with the global sampling, we were just happy for anything, to be very frank. And that's why there were locations that had a single sample, whether that was from a museum accession or from someone who could get just one. So we don't know that we're really picking up all of the genetic diversity in any one location. What I would say is that because of all the cities that we sampled, and especially looking at the co-ancestry maps and what they were showing, is that we saw that a little bit in the clustering analysis too, that every city has, even within the proportions of lineages, has a fairly unique and relatively uniform signature. And so I don't think there's anywhere we sampled where a single city was coming kind of out in two places.
But Emily, if you wanted to, and you did a high density whole genome sequence, even from a single individual, since you have two haplotypes, you can do the haplotype analysis and figure out what the effective population size was from which it was drawn.
Right.
So there's quite a bit you could do in addition if you just had whole genome sequence for those samples that you presumably currently have in your freezer. Is that true, or?
Yes, you're correct. And that's certainly the approach that we took with the demography and the MSMC, PSMC results.
Do you have those samples here in Memphis?
No, they're still with Jason.
Get them to send you an aliquot.
Okay.
Obviously it would be collaborative, But even those samples, it's not quite what I had in mind. I would love to go up a level in the taxonomy instead of just noodling around with just Rattus norvegicus, although that's fine, wonderful. But it'd be fun to do an entire genus of rats. That would be a game changer. Nobody has done that even for mouse. So we have to put the mice people in their place.
Okay, we can work with Ro and take a nice trip down to the Wappen region.
Well, if there's no more questions, I think there's a few people that wanted to chat with Emily. So unless you have a burning question, you can go off and have a great weekend. And then the people that want to chat, stay here and we'll get some more knowledge before we have a good meeting.
And we stay on the same link here?
Yeah, we'll just stay here and then let people who want to wander away zoom out.
Really encourage Anne, anybody from RGD, be wonderful to have you stay on.
Yeah, this was fascinating. In fact, Emily, I have to tell you that we had a journal club and one of our team presented your paper. Yeah.
Oh, thank you.
So when Hal forwarded this, I was excited to see this.
Thank you.